Are you safe there? Please be safe at home. What about last day's class? We discussed about 5 kingdom classification. The limitations of that 2 kingdom classification teacher is not saying again and again because you studied, I think. But in general, that 5 kingdom, which are they? Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, Kingdom Animalia. Who classified? R.H. Whittaker. Based on what? Cell structure, thallus organization, mode of nutrition, mode of reproduction, phylogenetic relationship, meaning evolutionary history. That much you studied, I think. You are very good students. You are studying. Then, what about the Kingdom Monera? Out of these 5 kingdoms, Kingdom Monera are unicellular prokaryotes. You know very well now what is a prokaryote. Coming classes, teacher will not say what are the important features of prokaryote and eukaryote. One more time teacher will say, that is because these monerans are microscopic prokaryotes. There is no true nucleus. Membrane bound organelles are absent. Genetic membrane, that is genetic material, is naked and coiled attached to the plasma membrane. These are the 3 important features of a prokaryote. Okay. Next kingdom we will study, they are eukaryote. Protistans are unicellular eukaryote. True nucleus present, membrane bound organelle is present, genetic material is enclosed in the nucleus. Okay. Today teacher is taking about Kingdom Monera. That is today's learning objective, structure of a bacteria. Okay. Why I am saying bacteria? Bacteria are the sole members of the Kingdom Monera. Okay. Kingdom Monera, the main item, main organism is bacteria. They are the abundant microscopic organism. Clear, you know. If we are taking a handful of soil, we can see hundreds of bacteria. Where we can see the bacteria? Bacteria we can see in the air, in the water, we can see in desert, we can see in deep ocean, we can see in hot spring, we can see the benthic level of ocean. Any places we can see bacteria. Some bacteria we can see in our body also. You know that some bacteria present in human intestine and also animal. Okay. One by one teacher will take. First we can see the structure of a bacteria. This structure of bacteria is needed. Again, chapter number 5, we will study in detail about a prokaryotic cell, which are the different parts of a prokaryotic cell. At that time also we are discussing about the bacteria in detail. Okay. For that portion also I need the structure of bacteria. Again, the next year you will study in detail about biotechnology. Biotechnological experiments also, this bacteria structure is very important. In the record also you want to draw this diagram. That is why I am giving this diagram. This diagram is not in the NCERT textbook. Okay. So very carefully take enough space and you draw the structure of bacteria. After that, I will discuss about the other topics. Okay, you come with me. This is the structure of a bacteria. This is Escherichia coli. You heard this term many times. That is E. coli. that is in short. This E. coli is the scientific name of this bacteria. This is E. Escherichia is the genus name. Coli is the species name. Genus name, first letter we are writing capital. Species, first two letters are small letter. You studied, I think, in zoology. So this is the genus name. This is the species name. Escherichia coli. Short form is E. coli. E. coli bacteria present in human intestine and also animals. Ready? Okay, we can see the structure. Don't think bacteria are very minute. They are microscopic. Don't underestimate bacteria. Its structure is also complex. Its metabolic activity also extensive metabolic activity we can see in a bacteria. Okay. So here we can see the cell wall. First we can see the cell wall. I told you the chapter 5 while studying cell we will study in detail about prokaryotes. At that time more detailed way we will study about this cell wall. Cell wall is complex in nature. Some other things are also present. That words are not mentioned in this chapter, so I am not saying. Now you have to remember this is cell wall. Inside it is the plasma membrane, that is cell membrane. This is the cell membrane or plasma membrane. Okay, and this plasma membrane is folding inwards. Can you see this? Plasma membrane is folding inwards. That is called mesosome. This mesosome is having many functions. Later we can study. Remember the term mesosome. So first it is cell wall. Inside the cell membrane. The cell membrane is also called the plasma membrane. Plasma membrane folding inwards. It is called the mesosome. Again, we can see inside cytoplasm the small vacuole present. Again you can see small mRNA. What is mRNA? Messenger RNA. And here ribosome. Why you are not saying ribosome also an important cell organelle? It is doing the process of synthesis of protein. Very important cell organelle, ribosome. Okay, this corona time, you know, this corona time, how this virus will act, you know, we studied. When viruses enter into a body, human body, virus is not having any cell, acellular. Later we will study. It is having a protein coat and a genetic material. When the virus is enter into a host or human body, the ribosome of that particular body is starting to the production of protein for the virus. Ribosome is very important cell organelle. Here ribosome present in prokaryote. What is the reason? Ribosome is not having any boundary. That is, it is not coming under membrane bound. Simply teacher will show you know. If it is a ribosome drawing, you have to write on this outline only. There is no correct membrane like this. It is wrong. Ribosome is not having any membrane. So ribosome is present in a prokaryote, that is in a bacterium. Again, this is the genetic material. Genetic material we studied, you know. Here there is no nucleus. You can't see any nucleus because If the true nucleus is absent, so the genetic material is naked and coiled attached to the plasma membrane. Clear, you know? This is the genetic material of bacteria. It is naked and it is coiled. It is attached to the plasma membrane. This is bacterial chromosome. But here there is no nucleus. Clear, you know? One important feature of prokaryote genetic material is naked and coiled attached to the plasma membrane. There is no true nucleus. Membrane bound organelles are absent. Okay. The next one here teacher marked, you know, plasmid. This is very important in the field of biotechnology. What is the importance? Have you heard anything about biotechnology? This is plasmid is extracircular chromosomal DNA present in addition to the genomic DNA. What's the meaning of genomic DNA? Genomic DNA, that is the bacterial DNA already present here. In addition to the bacterial DNA, there is another DNA there. This is circular. This is extracircular chromosomal DNA. In addition to the genomic DNA, it is having so many roles. Normally, if you are in the class, you know, teacher will explain all these things in detail. Simply, teacher will show. Suppose this is a plasmid. Plasmid is circular in shape. This plasmid is acting as in the biotechnology field, you know, this is acting as a vehicle or a carrier. This plasmid, this part is cutting with the help of an enzyme. I am not saying the enzyme name. Later we will study. So with the help of one enzyme, some part of this plasmid is cutting, and there is maybe a DNA. Biotechnology, we are dealing with manipulating genetic material. Here one DNA there. Remember, suppose this DNA having the insulin secreting gene there. Okay. Actually, I didn't plan explaining this, but while saying this plasmid, this also I want to say. This gene, insulin secreting gene, I want to put it here. So with the help of another enzyme, this insulin secreting gene can insert into this plasmid. I told you this plasmid is a carrier or a vehicle. It is carrying or it is taking this gene where it is needed, to which place it is needed. So it is acting as a vehicle. So after inserting one gene, you know, that the cell is working according to the wish of that gene. So in the field of biotechnology, this plasmid is very important. That much only you have to remember in this class, you know, about the plasmid. Later you will study in detail. So plasmid is extracircular chromosomal DNA present in addition to the genomic DNA. Okay. Then here for movement flagella present. You have to draw this diagram neatly. Cell wall, cell membrane, mesosome, mRNA, messenger RNA, ribosome present, small vacuole present, and plasmid present, flagella for movement. This is the bacterial chromosome. Neatly draw. Okay. So now we can see different shaped bacteria. There are mainly 4 shaped bacteria. Smaller classes you studied about that. Which are they? Coccus, Bacillus, Spirillum and Vibrio. I think you surely studied. We can see the picture. Coccus. Coccus, it is spherical in shape. It can see in singly, in pairs and in groups. Either one or two or in groups. Coccus, spherical shape. And one example, that is Streptococcus. Clear, you know? Streptococcus, that is the example of a coccus bacteria. It is either in single or in pairs or in groups. Teacher is giving something more. Not depend only NCERT. No. Teacher will give more topic because this topic is very important. Entrance examination and the coming years, even when you are going for degree courses, biotechnology courses, medicine, whatever it may be, all these portions are important. So, something more teacher will give. Coccus is spherical bacteria. Streptococcus is example. Then Bacillus, that is rod-shaped bacteria. This also we can see in single or in pairs or in groups. The example is Streptobacillus. Then again it is spirilla. This spiral-shaped bacteria. It is having flagella and it is the example Spirochetus. Example is not in the NCERT. The correct diagram you have to below write down the example also. Then Vibrio, comma shape. Vibrio, comma. Clear? There are 4 types of bacteria, depends upon the shape. They are Coccus, Bacillus, Spirillum and Vibrio. This is spherical, this is rod, this is spiral, this is comma shape. Clear, I think. Then we study about the nutrition of bacteria. So when the 5 kingdom classification explaining time teacher told you, I will take the portion later about the nutrition. Okay? Teacher is rubbing this because I need the space. There are different. You studied about autotrophic and heterotrophic. What is the meaning of autotrophic? Plants are mainly autotrophic because they can trap the solar energy and they can prepare the food material. Okay, that is solar energy converted to chemical energy. All the things you studied in lower classes. The case of nutrition, in the case of bacteria also, some are autotrophic. Again, autotrophic, they are not having chlorophyll. Then how they are? Photosynthetic autotrophs. What is the meaning of photosynthetic autotrophs? These type of bacteria can prepare their own food material from inorganic substances. In presence of light. Clear, you know? For autotrophic, photosynthetic autotrophs, that's the name. Photosynthetic autotrophs, they can prepare the food material from inorganic substances in presence of light. One another type, that is chemosynthetic autotrophs. What is the meaning of chemosynthetic autotrophs? They can also prepare the food material from inorganic substances with the help of chemicals. So mainly two type of bacteria are the photosynthetic autotrophs. There is no chlorophyll. Heavy wind is there. That is why the disturbance like that. Okay, strong wind is there. Photosynthetic autotrophs, they can prepare the food material from inorganic substances in presence of light. The other one, they can also prepare the food material from inorganic substance in presence of chemicals. Okay, the sentences are not in the textbook, word only mentioned in the textbook. The next one, nutrition, is saprophytic. They are saprophytes. What's the meaning? This also you studied. Saprophytic bacteria live on dead remains and they are absorbing food material from the dead organism. So saprophytic nutrition. Saprophytes bacteria, this type of bacteria live on the dead remains and they are absorbing the food from this dead food material, dead organism. So saprophytic. Some are parasitic. What is the meaning? Parasite. Some of the bacteria are living the host, 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 and they are absorbing food material from the host. This Escherichia coli, I told you, they are living in our own intestine. They are taking food from our own body and they are causing disease also. So parasitic bacteria are living in the host of other organism and they are causing diseases. Parasitic. One another type of bacteria, they are symbiotic. You know the meaning, symbiosis. What is the meaning of symbiosis? Both partners are benefited. You studied this, some type of bacteria named Rhizobium bacteria living in the root nodules of leguminous plants and they are giving the unavailable nitrogen is absorbing by this Rhizobium bacteria, give it to the plants. And in turn, this plant is giving shelter and food material to that bacteria. So both are benefited. Such a mutual relationship we can call symbiosis. So these are the different type of bacteria related to their nutrition. Very important topic, nutrition. One is, that is photosynthetic autotrophs, chemosynthetic autotrophs, saprophytic, parasitic and symbiotic. So if you are writing systematically this way, the nutrition, I think the NCERT is not at all mentioned, the word only mentioned. So if they can prepare their own food material, we can call them autotrophs. Otherwise, these saprophytes and this parasites, etc. They are depends upon others. Which word we are using? They are heterotrophs. See, teacher is preferring, write down point wise. Nutrition, heading. What are the type of nutrition? Which are autotrophic? Which are heterotrophic? That is my style. You have to follow. Surely you will get a full mark. Then the next topic. Actually, teacher planned to take about archaebacteria also. I don't know, the video is lengthy. That is archaebacteria. Under kingdom Monera, we are studying about bacteria, archaebacteria, eubacteria and mycoplasma. Then only kingdom Monerans over. Archaebacteria, speedily teacher will take and the teacher will conclude that topic. Have you heard about archaebacteria? They are primitive bacteria. Bacteria present everywhere, but these are special type of bacteria, this archaebacteria. They can survive any extreme environmental conditions. This bacteria can survive any extreme environmental conditions. Which are they? This type of bacteria are three types. First one it is thermoacidophiles. Thermo word meaning temperature, acid also These are one type of archaebacteria. They can survive high temperature and acid. Okay. Such a type of bacteria are called thermoacidophiles. Okay. Later, biotechnology portion studying time you will study about one bacteria that is called Thermus aquaticus. It is the name of one archaebacteria. Later we will study artificially the DNA preparation, that is PCR, polymerase chain reaction. Later next year you will study. At that time we need some enzyme. That enzyme is taking from this bacteria. Normal type of bacteria can destroy around 25 degrees centigrade. But this type of bacteria, I told you, it can survive high extreme environmental condition around 100 degrees centigrade. This type of bacteria can survive. Clear? What is the example? This Thermus aquaticus. That is one type of archaebacteria. Another one that is halophiles. Halophiles. Halophiles meaning high salt content, high saline water. This type of bacteria, archaebacteria, can survive in high salt content or high saline water. Okay. Another extreme condition this bacteria can survive. The third one, it is methanogens. Have you heard about the methanogen? You all surely heard about the methane gas, biogas. How this biogas is preparing from cow dung. So, these are called methanogens. These methanogens are one type of archaebacteria. They are living in the gut of ruminant animals. Which are the ruminant animal? that is cow, buffalo, etc. are ruminant animal. So these methanogens are present in the gut of this ruminant animal. They are digesting the cellulose, and from this cow dung, methane gas is producing with the help of this archaebacteria methanogen. So we studied about three types of archaebacteria: thermoacidophiles, halophiles, and methanogens. Okay. This I told you, normal type of bacteria can destroy about 25 degrees centigrade, but these can survive any extreme condition. Even hot spring also they can survive. High extreme desert, hot spring, deep ocean, saline soil, anywhere this bacteria can survive. These group of bacteria are called archaebacteria. How they can survive? Normal type of bacteria, cell wall is made up of peptidoglycan. Normal type of bacteria cell wall is made up of peptidoglycan. But this archaebacteria cell wall is made up of protein and polysaccharide. Because of the presence of this protein and polysaccharide, these bacteria can survive high extreme environmental conditions. This is not in the textbook. You have to remember. Cell wall nature is not is different. That point is there in the NCERT, but this point is not there. Normal type of bacteria cell wall is made up of peptidoglycan, but this archaebacteria cell wall is made up of protein and polysaccharide. Because of that, this type of bacteria can survive any extreme environmental conditions. Okay, I am concluding because otherwise it may be a lengthy video. I am concluding today's topic. We studied about first kingdom Monera, and under Monerans we are studying mainly about bacteria. Bacteria present everywhere we discussed, and also this structure of bacteria teacher explained very well with the diagram. Again we studied different shaped bacteria. They are Coccus, Bacillus, Spirillum, Vibrio. With the diagram we studied. Again we studied about the nutrition. They are photosynthetic autotrophs, chemosynthetic autotrophs, and heterotrophs, saprophytes, parasites, and symbiotic bacteria we studied. And again we studied about archaebacteria, special type of bacteria. They are thermoacidophiles, halophiles, and methanogens. Okay, I think today's topic is clear. Have a nice day. Thank you.
If you have any doubts, don't hesitate. Just dial up 9495275807 or 9526989605. I'll be there for you 24/7 as a guide. And PS, I know this is Liza Sebastian signing off. Goodbye and wait for the next video. Cheerio.